Hi. So thank you for the opportunity to come and talk to you today about unambiguous reporting and reproducible real-world evidence. I'll start with my funding and disclosures. A lot of the work that I'm talking about today was funded by Arnold Ventures, as well as a public-private partnership between FDA and a consortium of pharma sponsors. And I've also been a consultant to a software company. So obviously, reproducible science is very important, and there have been concerns about reproducible research in many scientific disciplines. We've already heard about cancer biology. We're going to be hearing about psychology and economics, and there's also been some replication attempts in randomized clinical trial data. And a lot of these replication efforts have generally shown that there's room for improvement. But there hadn't been a similar effort in my field up until this point. So by way of background, I'm a pharmacoepidemiologist. So a lot of the work that I do focuses on the comparative safety and effectiveness of drugs and marketed medical products. This is post-market phase 4 type of research. And in this 2x2 table of different types of research, where the columns represent interventional, randomized, or experimental versus non-interventional or observational, and the rows represent primary data collection for the purpose of research versus secondary analysis of data that's already available. My field really focuses a lot on this lower right-hand quadrant, where we do real-world evidence using routinely collected data, and by which I mean data that's collected as part of routine clinical healthcare, your insurance claims data and electronic health records and that sort of information. And there's been a lot of recent interest in using this type of evidence to help support understanding when and how we can use that evidence to support approval of new indications for drugs that are on the market or meet other post-approval study requirements. And recently in the US, there's been legislation about that, including the 21st Century Cures Act and PDUFA 6, and what used to be called adaptive pathways in the EU. Because our field uses secondary data and it's observational, there's often a lot of complex design and implementation decisions that we have to make in order to minimize the bias in the analyses. And often these types of decisions are not communicated clearly, making it difficult to reproduce the evidence, as well as evaluate the validity of the choices that are used to generate the evidence. And this is important because the end goal is to be able to use this information to make a decision about the benefit, harm, or value of a marketed medical product. But in order to do that, we need to have confidence in the scientific approach. Are the methods valid? Is the evidence robust? And is it transportable to different populations? In order to have that confidence, we need to understand how the evidence was generated. And it's at this foundational step where we have a hurdle to get over. Because of the lack of clarity in reporting, it makes it difficult for decision makers to trust the evidence and use it. When we have high ambiguity about how the evidence is generated, we just can't assess the validity of the choices or whether or not it's relevant to the question that we're trying to address. So when we have unambiguous reporting, not only do we understand how the evidence is generated and we're able to evaluate validity, we're also better able to understand why there may be reasons for divergence in evidence. And for my field, one of the issues is that credibility has been suffering when we see apparent divergence between multiple database studies that seem to be investigating the same question, as well as between database studies and randomized trials, notwithstanding the efficacy effectiveness gap and differences in inclusion-exclusion criteria. So before continuing, I just want to briefly define what I mean by reproducibility and replicability in the context of database studies, because there's many different types. The first being analytic or computational reproduction, meaning that you can rerun the same code on the same data source to recreate the same analytic population and run the same analysis to generate the same results. And ideally, you should be able to push the same button and get the same results. And this is an important type of transparency, but it's an important type of reproducibility, but it is not in and of itself transparent, because it can require literally thousands of lines of code to create an appropriately temporally anchored analytic cohort from the source data. And without clear natural language descriptions about what scientific decisions are being implemented, reviewers and decision makers can have difficulty determining whether or not they agree with the validity of those choices. The second type of reproducibility is direct replication, involving the ability to independently implement a specific study using the same data source to recreate that same analytic data population and apply the same methods. And this is an important type of reproducibility because it requires unambiguous reporting about the design and implementation decisions. And if the recipe provided is clear enough, there's no reason to expect a difference in result from an independent implementation of the same study. So our ability to independently directly replicate is a proxy for clear communication about how the evidence was generated. And we need that clarity to assess validity. The third type of reproducibility is conceptual replication or robustness, meaning that you're implementing a study asking the same question, same exposure, comparator, outcome, and estimand of interest. You could be applying the same methods in a different data source or applying slightly plausible alternative methods in the same data source or a combination of the two. And this is arguably the most common and most interesting scientifically, but when you have results that diverge, the question is why? And for that, again, you need clarity to understand, because it could be really subtle implementation differences in the design, in the data, or in the population. So there's already a lot of existing guidance about reporting for my field, including from the FDA, the EMA, research societies, and others. And there's some general principles that are common to all of these, including providing a detailed study summary that describes critical elements, having clear operational definitions for exposures, outcomes, and other measures. And it can be helpful to use a diagram to illustrate key aspects. But it's difficult. What are the critical elements, and how do you summarize them concisely, and what is a clear operational definition? What does that look like? So two research societies worked together a couple of years ago. They put together a joint task force on real-world evidence for healthcare decision-making. And there were two outputs of that. The first focusing on transparency in the process of conducting database studies, or what did you plan to do? And this was really focusing on pre-registration and that sort of thing. The other was focusing on clarity of study execution at the beginning. What did you plan to do in detail? And then at the end, what did you actually do in detail? And what were the changes along the way? So focusing in on this second paper for a moment, what we try to do here is create a catalog of operational parameters and design decisions that an investigator is making when they're implementing a study using a healthcare database. And unlike the existing guidance, which by their nature tend to be a bit general to cover a broad base, we tried to be very specific about the decisions that are made when you're moving from the source data, which is longitudinal streams of healthcare data anchored in calendar event time, and all the data transformations that you perform upon it to move into an analytic population, which is appropriately anchored in patient event time. We followed up with a paper that developed a framework for graphical depiction of study design using these type of data, and this framework was designed to try and clearly show the temporal anchors and steps used to define an analytic cohort in a way that minimizes misinterpretation from vague prose. So here's just a quick, very simple example of what a design diagram might look like. It's designed to be read from top to bottom, indicating the steps that are taken to create the analytic cohort. This is a study looking at the risk of angioedema with ACE inhibitors versus angiotensin receptor blockers. So you've got the first vertical line is the date in which you enter the study population. This is day 0, and all other windows are indexed against day 0. So you can have windows. You can look at the widths of the bar to indicate how long they are, as well as there's the days indicated in brackets relative to day 0. So you might want to have prior enrollment requirements, a washout window to make sure that you have incident exposures and incident outcomes, apply exclusion criteria for age as of the index date, have covariate assessment windows of varying duration and relation prior to or including the index date. And this is where you try to measure the confounders, because this is observational data. There may be reasons why you get one drug versus another that are related to the outcome of interest, and those are the things that you want to adjust for. But you need to be clear about how you're measuring the things that you're adjusting for. And of course, then you have your follow-up window, which may include day 0 or not, and be censored upon different criteria which are indicated in the footnotes. So this is a very, very basic design diagram, top to bottom, the steps that you're taking to create your analytic cohort. Just to walk in a little bit more depth about one section here, when we talk about defining exposure definition, having clear operational definition of that, we don't just say we're studying ACEs versus ARBs. We really need to get into a little bit more detail about that. So you might want to talk about the type of exposure. Is it incident, cumulative, or time varying? Items such as whether or not we want to consider including an induction window for exposure effect if we believe that there's biologically no reason to expect an effect for, say, 6 months or a year, then maybe you want to build that into your exposure risk window. Additionally, sometimes we're going to see things in the data where we have multiple dispensations plus days supply where you have overlapping days, and how do you handle that? Do you ignore the overlapping days? Do you tack it on at the end? Or what if you see multiple dispensations with day supply and there's a gap in between? Do you censor at the end of the first day supply? Do you bridge the gaps if they're less than, say, 14 days? And if you do that, then for consistency, do you tack on the same amount to the end? And these types of detailed decisions will determine which outcomes are going to be counted in your analyses. and often are not reported in clear detail. And there's other things in here that I'm not going to go over, but this is the level of detail that I'm talking about. So the project that I will be particularly focusing on is trying to measure where we are now and how can we improve in terms of clarity of reporting, reproducibility in terms of direct reproducibility, and robustness and transportability to different populations, with the idea that we can use this information to work with key stakeholders, develop empirically based standards and processes to improve those characteristics for our field. So what we did to do this was we started with identifying a random sample based on a Google Scholar search for peer-reviewed publications, focusing particularly on descriptive or comparative effectiveness or safety studies. They had to be conducted using healthcare databases, particularly one of 4 that we had licensed access to. These are often used for research purposes. They were MarketScan, Optum, CPRD, and Medicare. We evaluated the clarity of reporting for our sample of 250 studies using a standardized extraction form based on the ISPOR-ISB Joint Task Force Catalog to allow us to measure and describe how often different parameters were or were not reported. Then we took a subsample of 150 studies oversampling the comparative ones that we attempted to directly replicate using the same source data as the original authors to try and recreate their analytic populations and analysis. We used the same years of data as them and tried to use the same methods as best we could interpret, with assumptions when those were unclear. And our study team did this blind to the original results. We had one team member go through and redact measures of associations and blank out the entries in the tables. After attempting to independently replicate these studies, we contact the original investigators to discuss our assumptions and try and understand where we may have misinterpreted or made choices different than the original investigators, based on our interpretation of what they made publicly available in their manuscript, appendices, or citations. And finally, we are working on robustness evaluation to see how variable the estimates are when we implement different parameter choices or implement in different data in populations. So we're about 120 out of 150 studies completed right now. Here are some of our interim results. I'm showing you here in this figure differences in the binary and categorical characteristics of the cohorts in the publication compared to the replication. On the x-axis here, these are the different studies on the columns, and on the y-axis is the difference in prevalence of that baseline characteristic. Basically, we look at the table 1 for the publication and the table 1 for the replication and find the same value in each and take the difference. On average, the difference was 0, but as you can see from the figure, there's quite a bit of spread. One of the things that I should note is that some of them are colored blue and some of them are colored red. The blue ones are covariates where the code algorithm was provided. The red ones are where the code algorithm was not provided. And from the spread of these, you can see that that doesn't really explain why we have some of these differences. And that's partially because how we measure the covariate is not just about the code, it's about which care settings you're looking at, what diagnosis position are you using, the timing over which you're measuring the covariate, and sometimes you would make assumptions about those even when they provide the codes for the algorithm. Furthermore, covariate codes were provided more often than the code algorithms used to define inclusion-exclusion criteria. So if we had to make assumptions about the exclusion criteria, we could be trying to measure the covariates in a subtly different population than the original investigators. Just as an anecdote of why sometimes we see really large differences in these baseline characteristics of the replication cohort using the same data source to create that analytic population. This one is a study where the authors provided a citation to a comorbidity score, and in that comorbidity score was tumor and malignancy as one of the components, and it had a weight of 2. So in our replication, all of our patients had a score of at least 2 because tumor and malignancy was also part of the inclusion criteria. But when we unblinded the table, we saw that in the original publication, 75% of patients had a score of 0, suggesting that perhaps the authors didn't count the weight or they did something else that we just didn't know about because it wasn't reported in their methods. So going on to the metrics that we use for looking at effect sizes and confidence limits, some of our primary measures of interest were looking at the difference in the log effect size, as well as the calibration of the replication against the publication with a correlation coefficient. We also looked at some other descriptive measures, primarily because they have been used in other replication studies, but they have some limitations, particularly in our context. So one of these is a binary measure of agreement, looking at whether counting as agreement if the replication had an estimate on the same side of null as the original, with the p-value on the same side of 0.05. And for our setting, this is it has limitations partially because we often have really small effect sizes in real-world evidence, and it's easy with small differences in implementation to bump from one side of the null to the other. Additionally, even if we exactly hit the point estimate with our direct replication, if the original publication had a p-value of 0.04 and we had a p-value of 0.06, using this binary measure of agreement with the arbitrary cutoffs, they would be counted as non-agreement. We also looked at confidence interval overlap, which had similar limitations. Again, using real-world data, we can often encounter situations with really tight confidence intervals because it's quote-unquote big data. So even if we have a clinically meaningless difference in effect size, sometimes the confidence intervals are so tight that there can be very little overlap. So keeping those caveats in mind, here's some of our interim results. I'm showing you here the difference in the log effect size between the publication and the replication, where we saw the average difference in that log effect size was 0.05, which is actually pretty good. We're aiming for 0, and that's not too far off. But you can see that the spread is quite wide, ranging from a relative effect size of 50% to 350%. Here's a calibration plot of the effect sizes in the publication compared to the replication, where what you're aiming for is a diagonal line indicating that the estimate in the publication is equal to the estimate in the replication. What you can see is that the estimates tend to follow that diagonal, and we actually have a pretty strong correlation coefficient at 0.74. However, we have some of these studies that, despite our best attempts to replicate using the same data source and using the same methods as best we could interpret, that we just weren't able to replicate very closely. So here, again, some anecdotes. This is a particular study that was looking at a hazard ratio comparing two drugs on risk of major bleeding. And in the original publication, they had a hazard ratio of 1.9. In our replication, we had a hazard ratio of 3.4. And some of the notes from our replication team, for each of these studies that we do, we have a sheet of assumptions that we note down as we're going through. We had to make an assumption here, we had to make an assumption here. But here are some of the ones that we did for this study. In the manuscript, there were discrepancies in the text and the attrition table about which exclusions were applied. So we had to make a decision about which ones to actually implement in our replication. We also had to make assumptions about how inclusion criteria and covariates were defined, what codes were used, what care settings were looked at, what was the diagnosis position, what was the timing. Was day 0 included in the assessment window? And this can often be influential in these types of studies. The authors did provide the outcome code algorithm in the appendix. When looking at the characteristics of our replication cohort compared to the original, our cohort was about 10% larger, and most of the baseline characteristics were within 10 percentage points of the original, which 10% might be a little bit large still, but they were within 10%. But we noticed that despite being relatively close in being able to match the baseline characteristics of the cohort, the outcome rate in the original publication compared to in our replication compared to the original was very different. Now, what they had reported using for their outcome algorithm was a set of codes in the primary position in a primary inpatient setting. And we did that, and our rate of events was much lower than theirs. So we thought maybe they made a mistake in reporting, maybe they did any inpatient. So we tried that, and our results actually looked a lot closer. So that may be a potential explanation for why we had such differences here, but unless we can get confirmation from them, we don't know what the actual reasons for the differences are. Here's another anecdote example, also looking at two drugs comparing a hazard ratio for bleeding, where the original estimate was 1.2 with the confidence intervals excluding null. The replication was a 0.8 with 95s including null. And we again had to make assumptions about how exclusion criteria were defined in covariates. Again, they provided the outcome algorithm. However, they weren't clear about defining the exposure risk window and which outcomes were counted, because we had to make assumptions about censoring criteria, how exposure stockpiling, bridging, and extensions were made. And obviously, as I mentioned, that determines which outcomes are counted. And our assumptions were probably a little farther off this time because our replication cohort ended up being 30% larger than the original, and a lot of our baseline characteristics were very different, again, from the original publication. So here are some of the metrics that I mentioned before, the descriptive metrics that have some limitations. And I'm presenting two columns here because depending on how you frame it, it can sound better or worse. So I'm going to focus on glass half full now. Using the binary measure of agreement, 63% of the estimates were on the same side of null and had p-values on the same side of 0.05. when we're looking at confidence interval overlap, 85% of the confidence intervals had any overlap, and if they did, about half of the confidence limit range was shared. We are planning on doing further evaluation about what's driving this variation between the publication and the original, given that we're trying to implement it using the same source data to recreate the same analytic population. And some of the differences or factors that we're planning to look at include clarity of reporting on specific types of parameters, looking at the journal impact factor and the type of journal. Is it a specialty clinical journal versus like an epidemiologic methods journal? Funding source, author experience, proxied by citation impact factor, and time. Are we improving over time? So I mentioned before that after attempting to independently directly replicate studies, we're reaching out to authors to see if they will talk to us and help us understand where some of our assumptions may not be lining up with what they had done. So out of 49 attempted contacts thus far, about 33% have responded. Among the people who have not responded yet, we consider 40% non-responders after 3 attempts at contact. And 21% of our emails for the corresponding authors are undeliverable. And 40% we're still trying to reach. So among the responders, I think we had similar patterns as Tim mentioned. These are just themes, not direct quotes. We have people respond back just to say, I don't want to participate, or essentially leave me alone. We have people respond to say that we will respond after carefully reviewing the document and then don't respond to our queries after that. And then there's a special population, very select, who say, like, I forwarded it to the study team. We'd like to schedule a phone call, and we'll discuss your assumptions together. And these calls are always very helpful. They're like, this is the algorithm we actually use. Here's a code snippet. Oh, well, we didn't do that, and that sort of thing. And so this is a very special and select population. Anecdotally, it seems like the people who are doing this tend to already have studies that we were pretty close with replication, and they're just really eager to help us get even closer. Finally, we're also working on conceptual replication, trying to systematically quantify robustness and transportability by examining sources of variation. And we're doing this by defining a research question and then designing a matrix of study specifications based on what we see in published papers, based on plausible parameter variations from the Asporius-B Joint Task Force catalog, and then implementing the entire matrix in multiple data sources. This ideally would allow us to describe the magnitude of differences both within and between data source, and also to do some regressions to quantify the drivers of variation. And in this figure here on the right, this is just from the repeat replication sample. We had 9 papers looking at this question of the bigatran versus warfarin, these are anticoagulants, on risk of major bleeding in patients with atrial fibrillation. So that's the question, these are the results. And you can see that there's quite a bit of a range in them, even though many of these were implemented using the same data source. Obviously, there were differences in parameter choices between studies, and including there were some studies, or one study that there were some really clearly flawed design choices. But it's in this phase that we plan to systematically examine what's driving variation, trying to evaluate how much of it is random noise, how much is due to systematic errors, and how much is due to effect modification. So some concluding points. There is room for improvement, but I think we have some ideas about how we can get there. In particular, exclusion criteria is one area where we're often underreporting. It's just mentioned in passing without any real clear definitions about how you're defining how you get to your study population. Temporality is another big area where details are sparse, particularly when it comes to the window for outcome ascertainment, because often they provide the outcome codes. That's very frequently provided, but how follow-ups start and end is often unclear, especially when studies are reporting censoring on exposure discontinuation. And for those studies, we often had to make a lot of assumptions, and obviously those assumptions affect which outcomes are counted. And it's really hard to replicate an analysis result if you can't replicate the analytic cohort. Another take-home point is that our replication results are about clarity of reporting and not necessarily an assessment of validity in and of itself. We had studies in our sample which we noted there were clear design flaws, but because they reported what they did so clearly, we were able to replicate the flawed result. We also have studies in our sample that may have been really great studies, but we couldn't tell what they did. So we had to make assumptions, and those assumptions may not have aligned well with what they actually did. So as a first step, we're aiming to have unambiguous reporting about how the evidence is generated so that we can make this determination about validity. Because without that level of clarity, we're left in a situation where we cannot differentiate between the high-quality studies whose evidence we want to use to inform our decision-making from the low-quality studies whose evidence we may want to disregard. There's some parallel efforts in progress, all under the guiding principle of don't let perfect be the enemy of good. And one of these is a multi-stakeholder steering group involving ISPE, ISPOR, Academy Health, Duke Margolis, and National Pharmaceutical Alliance, really working together on trying to encourage registration of hypothesis evaluating real-world evidence to try and increase the transparency of the research process. And it's really focusing on practical steps and logistical barriers to doing so, because the existing sites like clinicaltrials.gov and EUPass aren't really designed for this type of research. Additionally, we're working with FDA as part of a public-private partnership on developing a structured reporting template with the detailed steps and design visualization to try and increase clarity in reporting about study implementation, as well as simplify reporting and trying to maximize efficiency, both from the researcher perspective as well as the reviewer perspective. I'd like to end by thanking the Repeat core team that's been working flat out for the past couple of years on these studies, and also our large scientific advisory board, including regulators, health technology assessors, delivery systems, patients, payers, industry, journal, and research society, all of whom have been providing invaluable feedback from the beginning and hopefully helping us move toward shifting the way we do things. That's it. Hi.
Hi. I'll model the new questioning template. Steve Goodman, Stanford Medicine and Meta Research and Epidemiology. So this is great work, and your team are to be congratulated. I just wonder how it relates to the work that I'm sure you know well from OMOP and Odyssey that tried to do something similar to this on a massive sort of machine learning type scale, where they looked at similar questions across multiple databases, adjusting all the design parameters, and also calibrated these using negative controls to look at the biases. And they're very, very critical for some of the reasons I think that you pointed out of what they would call bespoke analyses, that is, analyses that are curated on one data set with very careful calibration of all these parameters, and they look at the variation of results according to all these, many of the things that you talked about, and again, with negative control. So how do you juxtapose this and contrast these results with those of OMOP, which looked at, I think, hundreds and hundreds of drug outcome combinations?
Right, so my understanding of what OMOP or Odyssey did is they basically tried lots of different study designs for a set of positive and negatives, quote-unquote known positive and negatives, regardless of whether or not those study designs were good for the question that was being asked. And actually, Susan Gruber went through and winnowed down all of the combinations that they looked at, and really there was only a handful in there that would even be reasonable to attempt from a scientific perspective. So if you want to throw in, at the risk of sounding too critical, garbage at it, you will get garbage out. So really thinking about the scientific decisions and picking thoughtfully what designs you want to ask for which questions is more where I'm coming from, as opposed to trying all by all, even if it doesn't make sense.
How would we know a priori? So I haven't seen her work, but she's making a judgment as to which designs are appropriate and which designs weren't. They obviously didn't necessarily think that it was that obvious that the designs were that inappropriate. What are the features that would make a cohort design of using certain induction period or certain exposure level obviously completely invalid?
Well, I wouldn't say necessarily a cohort study, but included in some of their examples was, I believe, something like NSAIDs and gastrointestinal bleeding with the self-controlled design, where it clearly violates the assumption that the outcome not affect future exposure, and yet they did it. And the estimate was correct in that you got the correct positive, but the estimate was also biased because it was extremely inflated due to the violations of the assumptions. So that's sort of some of the subset of the comparisons that they did. You might get the right answer just by serendipity, but it's not necessarily an accurate answer.
Okay, thank you.
Hi, David Reinstein, University of Exeter economics academic. Sorry, this is a complete naive clarification question, but you said these were database studies. I'm not in medicine. I don't know what database studies are. Were you just taking the same data that they said that they used and running the exact same analysis as far as you could recreate what they wrote in the paper? In other words, you didn't collect any new data. This was just reanalysis using the techniques that they said they used. Is that correct?
Correct. So in my field, we often use large healthcare databases that are routinely used for research, even though they were not collected for research purposes. So these are large administrative claims data, and as well as integrated or not integrated electronic health records. And so from that, if you access the same source data, you can try and recreate the analytic population as long as you're able to license access and that sort of thing. You can recreate the analytic population and try and recreate the analysis as well.
So in principle, you should have gotten the exact same results. It's not just a resampling or rerun. And is that correct?
In principle, yes, if we understood to the finest degree what they actually did. Although there is a caveat that we may have been using different data versions than the original authors. The data vendors will put out different data versions. There should be very small differences, but there could be some of that which I don't think should affect it too much.
And just to find out, none of them could have just shared their code with you and say, just run this. That never happened? And then you would get the exact same thing as them, or?
They could have, I suppose. The question is not so much, does their code work, but do we understand what they did? Is that clear to us? And if it's clear to us what they did, it doesn't matter if they share their code because we should be able to do it ourselves.
Sure. Okay, thank you. I just want to make sure.
Yes, hello. Olavo Amaral from Federal University of Rio, Brazil. We have a big replication project in the lab sciences running locally. I want, can you give me an idea of like the amount of work that each of these replications actually take? Because I mean, I think one of the take-home points from Tim Arrington's lectures, like you can't do this every day, this is too costly and takes a lot of, but maybe for reanalysis, this could be, I mean, do you think this could plausibly be incorporated in the workflow, like regularly doing multi-site analysis or regularly having this in the review process, or does that just take too long?
I think it's definitely easier when you're using secondary data. So even outside the context of replication, if you're aware of the FDA's Sentinel initiative with their distributed data network, they send out packages and you expect a turnaround within 7 days of all of those results because everything's in the same format. For us, if we're not trying to come up with a recipe, it's a lot quicker to be able to do these analyses. For people who are trying to do novel or new studies, trying to pick parameters and thinking through things, it can drag it out a little bit. But in terms of our project, it's kind of unprecedented. But what we tried to do was aim for a 2-week cycle. 2 weeks to put together the protocol and 2 weeks to implement, and then 2 weeks to review with the faculty partner. There's always at least 3 sets of eyes looking at each study. But that was our cycle, and we sort of staggered it. So at any point in time, each team was working on at least two or three studies.
Thank you for your talk. I'm Zoltán Kekec, researcher at the Hungarian Academy of Sciences and ELTE and Lund University. So my question is about how widespread are registering these analyses beforehand, and did you find any pattern of higher reproducibility in registered database analysis versus non-registered ones?
We haven't looked at that yet. I think registration isn't very common in my field. I think it's only required for some EU regulatory-based required studies, and so for the rest of the time, people don't generally register in our field. So I think if it was more widely registered, we might be able to look at it a little more closely.
Hi, Nick DeVito, University of Oxford. I'm a doctoral student. So off of that, it's really interesting that there's not more pre-registration because this is probably the field most susceptible to like p-hacking and outcome switching and things like that. So I guess you already answered the question, but I just wanted to make that a comment, but I have a different question also. So when you found discrepancies and you reached out to talk to people and then they turned you down, they said they didn't want to talk to you, did you consider any more formal methods of, say, writing a letter to the editor or commenting on their piece? Or did you not want to do that for certain reasons?
Well, we're not trying to be adversarial here. We're just reaching out to try and understand. If they're willing to talk to us, they can help us move things forward. But we're not trying to point at anybody or poke fingers. It's really just about understanding, and we're coming at it very humbly because we could have misunderstood things. And that's how we're approaching them. Like, can you help us understand? We're just trying to understand what you guys did to get your evidence
So
without judgment.
Just to put back on that a little bit, like if you gave them the opportunity to talk and they turned it down, is it really being adversarial then to make use of the, accepted standard in science to then, put an official note in that you couldn't replicate? Like that's something people should know. This is unreplicable research. It just seems like it would be the standard way of doing something. I think it's not right to say it's adversarial after you gave them the chance to respond and they declined.
That's a fair point. Okay. Thank you.
Dorothy Bishop, University of Oxford, Developmental Neuropsychology. Really just following on from that question, I know when I've tried in slightly different contexts to contact authors, I get rather similar proportions, and I think other people have reported the same. There's a sort of division into people who don't want to do anything with you. Just to say, in my experience, it can help if you write to more than one author, because socially they're sort of having to collude to refuse you if you've got at least two of them on board. And I wondered whether you tried that or whether you only ever corresponded with the corresponding author.
So I did ask my team to look for first and last author if possible, but often all the email they had was the corresponding author listed in the publication. So if there was more information readily available, they did try. Otherwise,
And did you feel it made a difference to ask two people rather than one?
not that I can tell, but our sample size is still pretty small in
Okay.
terms of responders.
Just as a general advice, I think it'd be good to do a study on it, but I think it possibly might help.
Thank you.
Sure, this is the last question.
Okay.
Hi, Katie Corker, psychology. My question is about what you describe as discrepancies or differences between the original studies that you were trying to replicate. There's, I think, a variety of reasons for those discrepancies, and one obvious one is simple mistakes. So even if people don't respond, do you have a sense of what proportion of the discrepancies that you see seem to be due to mistakes or errors in reporting, or which are just simply inexplicable?
I know that there are some that were likely mistakes, but I don't know what proportion because I can't differentiate that from unclear reporting. So I know it's some part of it. I just cannot tell with the information that I have.